Good evening, everybody. Well, actually, I should really be saying good morning, good afternoon, and good evening. We are so pleased to welcome so many people from around the world to this very special post-ASH webinar. Now, I know that a lot of you have chosen the option, and this is the first time we've done it, to have the languages in your own language in subtitles so that you can hear the speakers. I think the problem may come that and we do want feedback on this, may come that some of the translation may be not as straightforward as we're hoping. But please bear with us because it is the first time we've done it, and we're very, very excited to do this. I have to say we couldn't do this without the wonderful clinical team we have, who are going to be giving us some really excellent talks. And the other people I want to say a special thank you to are our advocates organizations in Europe. Well, there's MPN Global and MPN Advocates Network, who've also spread the word to other patients to join this webinar. What gives me a huge thrill to say, and I'm just going to run through it because it is so exciting. We've got patients joining us from Argentina, Australia, Belgium, Canada, Cape Verde, Chile, Croatia, Czech Republic, Denmark, Finland, France, Germany, Ghana, India, Ireland, Israel, Italy, Jersey, Kuwait, Netherlands, New Zealand, Serbia, Spain, Sweden, Thailand, Trinidad and Tobago, and the USA. For me, this makes us a truly global community, and I thank you all. Now, if you're having any problems in getting on to the real-time translation, please put it in the chat. And then Debbie or Maz will try and help you sort the problem out. But I'm now going to hand over and say a huge thank you to Professor Claire Harrison, who is going to introduce you to the agenda and to the wonderful clinical team. And again, a huge thank you to them for giving their time to share their wisdom that they have learned and gained from ASH in December. Thank you, Claire.
Thank you very much, Nona, and hello, everyone. This is our agenda for this post-ASH webinar. You can see we're already a little way through the agenda. I'm super grateful to my lovely colleagues, Professor Beth Saylor from Oxford, Dr. Alessia Khan from Leeds, and my lovely, lovely colleague, Priya Sriskandaraja from Guy's and St Thomas's, for joining me this evening. We will do 4 presentations covering some of the highlights of the meeting, and then we will do a question session. Thank you so much for the questions that you've submitted already. We'll try to answer as many of them as we can. If you want to put questions in the chat, we will also look at them. But please do try to remember that we can't answer individual sort of medical queries. So first of all, I want to tell you what ASH, or the American Society of Hematology, is. In essence, it's an absolutely massive conference. You can see, I want to tell you what the American Society of Hematology on the slide here. That there were over 30,000 delegates that come from across the globe. I'm just going to ask everyone to mute if possible, because I'm getting a little bit of feedback. But here you can see an idea of how busy this Congress is. And you really need a sat-nav to get around, and you could be in this Congress and not bump into a colleague who does a different part of haematology. It is our go-to meeting for our community, and it's very busy. We typically are running around from 6:30 in the morning until quite late in the evening. And what are we doing? Well, we're hearing the latest news. We're exchanging views on that news. And this is what you'll hear Beth and Alessia and Priya and myself doing. We'll tell you what we heard, but also what we think about it. We have lots of discussion. We try to further collaboration, and we learn from other disciplines in haematology. And we try very hard to digest what is often a massive amount of information. So we submit information in the form of things called abstracts. These are 400-word summaries of new information. And this year there were over 8,200 different individual submissions. So it's very, very busy. This year at ASH was a very special year for MPN. So two of the absolute heroes in MPN, you'll recognise Ruben Messer, but you may not recognise Radek Skoda. They were jointly awarded the Ernest Buehler Prize for their work in MPN. Radek for leading scientific work in the field for many, many decades, and his was one of the groups that discovered the JAK2 mutation, but he's also done lots of work on familial and inherited MPN. And Ruben, for all of his work, working with the community in bringing therapies to the fore. And it's exactly 14 years since the launch of ruxolitinib, and where we were all at ASH and seeing Jakavi written everywhere across the meeting. So that gives you a flavour of what's to come. And then, Beth, I think I'm going to hand over to you to talk about one of the absolute top topics, which is targeting the mutated clones and the work that was done and presented at ASH. So over to you, Beth.
Thanks, Claire. And shall I share the screen from my side if you stop sharing? Right, let me just get the slides up. So hopefully you can see this coming up now.
You can see the end of your presentation now,
Sorry.
I think.
Yeah, let me just go back to the beginning, sorry. Perfect. Okay, thank you, everyone. So in the next 10 minutes, I'm going to give you some updates on agents which basically are selectively targeting the MPN clone. And what that means is these agents, these newer drugs, are trying not to touch the healthy wild type cells that still exist within the bone marrow and the blood of patients with MPNs. So the data shared at ASH was mostly focused on these emerging antibody therapies that are targeting mutations in CALR, calreticulin, but people on the call will know that basically MPNs can be driven by one of three mutations, the most common being a mutation in this gene or protein called JAK2, the JAK2 V617F. And a third of patients then have a mutation in CALR, and then around 5% of patients in MPL, and then some patients will not have an identified driver mutation. So to start then with the mutant CALR agents, just to say there are actually a lot of excitement about, there is a lot of excitement about agents targeting this protein, and that's because of its really unique and biology. So the mutation in CALR is what initiates the MPN in patients, and it's quite fortuitous from a therapeutic perspective that the product of the gene mutation that causes the cancer produces this abnormal protein that ends up sitting on the surface of the cell, which means it's available for therapies to selectively identify those cells and then try to clear them from the body. So the data I'm sharing today is all from what's called a monoclonal antibody. So an antibody that's basically doesn't have anything else added to it. It's just binding those cells and then preventing them from working as well. But there are also antibodies in trial that work a little bit differently that I'll come to at the end of the talk that are basically going after this target in different ways. But clinical data from those agents has not yet been shared. We're hoping that comes later this year. So the trial where we had 3 abstracts at ASH is on a agent called Inca 033989. So this is a first-in-class mutant CALR targeting antibody. This trial is open currently across the world, and we're open at 3 sites in the UK as well. So some people listening, I know maybe on the team that's only there in the mutant form, so it doesn't touch the healthy form of CALR. So the study's open for patients with essential thrombocythemia and also for patients with myelofibrosis at the moment, or when this data cut was done, it was for patients with myelofibrosis who've previously been treated with a prior JAK inhibitor, although some patients actually were not suitable for JAK inhibitor therapy, so they had never seen a JAK inhibitor before. So that's the patient group. So first of all, to share with you the data from patients with essential thrombocythemia. This is quite a busy plot, but I'll just talk you through it. So this is showing platelet counts in people with ET. So here's the beginning of the study, baseline, and you can see that these platelet counts are very high. They're around 1,000. So these dotted lines here represent the normal range. And then you can see after 1 dose and 2 doses of drug, the platelet counts are really beautifully normalizing into that target range. And then the blue and the green, what that's showing is, because this is an early study, some patients were getting extremely low doses of drug. And then once we knew that was safe, the dose was then increased to see whether you can achieve better targeting. To the patients, the bars in green are representing data from the patients receiving those slightly higher doses, and the blue ones are the very early doses that were tested. But actually in both groups, you can see this really nice normalization of the platelet count, and that's maintained then over what's shown here is the first 7 months of study, although some of the data then goes out to well over a year. So really importantly, I haven't put the data on the slide, but what was, I think, particularly impressive about the data that was shared was that over 90% of patients are actually still on the trial, and only 4 patients happened to discontinue the study, and that there were no dose limiting toxicities observed, which basically means that this is an extremely well tolerated drug with very minimal side effects. So that's quite unusual, particularly when testing agents in this first in human setting. So there were a couple of side effects seen, but really nothing that limited the amount of drug or the dose of the drug that was able to be administered to patients. So then going beyond platelet count, they also then looked at the percentage of cells in the body that carry the mutant CALR signal. So here this is looking at what's called the variant allele fraction, which is another way of really estimating the cells in the body that are mutant. And what you can see, so each bar here represents an individual patient, and you can see that most of these bars are going down. So over the course of that first 6 months, you can see that patients, almost all of the patients, in fact 96% of patients, had a really nice drop in the percentage of in the variant allele frequency, the burden of mutant CALR in the body, some of them going right down here to almost 100%. So that's very impressive, and I'll say that actually there aren't really any other agents that do this as effectively, particularly for patients with mutant CALR. Of course, some patients with mutations in JAK2 particularly, and also sometimes in CALR, know that interferon can also do this and reduce the mutant allele fraction, but that tends to happen actually over a much, much longer period of time and not as robustly as is being shown here. So then moving to patients with myelofibrosis. So the data shared was for a number of patients also in myelofibrosis. Again, here, each bar here represents an individual patient, and you're looking at the best change in the spleen size. And these dotted lines are showing really quite arbitrary cutoffs that we use to evaluate new medications tested in this setting. The most commonly used one is this minus 35%. So that represents a 35% reduction in the spleen size. So again, most of these are going down, representing really nice improvements in spleen volume, and many of them going really down quite a lot below this target of minus 35%. So I know you'll notice some of them going up. Now, one thing that's contributing to this is that many of these patients would have recently come off a JAK inhibitor prior to going onto the study, which means that coming off that drug might have led to this increase then in their spleen size. So that's the key message there. And then also looking at symptoms, again, a lot of patients here having nice improvements in their symptoms. So this is looking at the percentage change in symptom score. So again, going down is good, a decrease in symptoms. And some patients here, their symptoms are going away entirely, so 100% clearance of their symptoms. Then maybe this is slightly more unexpected. So you can see here, this is looking at anemia benefit in patients on study. So the blue line here represents patients with myelofibrosis who had anemia at baseline going into the trial. And what you can see is that over the course of the first couple of cycles, really impressive improvements in their hemoglobin, so resolution of anemia, and then the hemoglobin is then staying within the normal range. So this is nice because we know that, for example, treating patients with myelofibrosis with JAK inhibitors can often actually worsen anemia. There are some other JAK inhibitors, such as momelotinib, that some of you may be familiar with, that do lead to an anemia benefit. So it's really nice to see this also occurring with this newer antibody therapy that's much more targeted than the current JAK inhibitor therapies. And then finally, so there was a small number of patients who were treated both with this antibody and also with ruxolitinib. So these were patients who were already on ruxolitinib but hadn't had an optimal response. And then those patients were also tested. So what they were looking for here was to see whether adding in the antibody gave additional benefit. And again, they found that even in the setting when patients were still on ruxolitinib, the majority of patients did have improvements in their spleen volume, improvements in their symptoms. But actually, interestingly, here you can see there isn't really much anemia benefit here, which probably speaks to the impact of ruxolitinib in this setting. So they did some more translational work as well, so looking in more deeply really to try and characterize responses. And I've just included this slide because I know that many people have bone marrow biopsies, particularly when they're on clinical trials, and I thought it might be useful to show you how that data is used. So this here, if you look on the left, this is an image of somebody's bone marrow at screening. So before going on the study, and then here after 6 months, the mutant CALR. And you can see a really nice reduction then in the number of yellow cells here, suggesting that there's been really nice clearance of the MPN from the bone marrow. And that was seen across patients with ET and also patients with MF. And interestingly, while the total number of these megakaryocytes, which are really increased in MPNs, goes down, and the number of megs which are mutant goes down, the number of megs which are wild type, so the healthy megs, goes up, suggesting the bone marrow is recovering. So I mentioned there were other approaches on the horizon, and I don't have data to share because that data hasn't been publicly presented yet. But some of you will know there is also another study being run by a different sponsor of a different kind of antibody that works a little bit differently. It binds to those mutant CALR cells, and then it draws in the body's own immune cells, the T cells, and tries to get the T cells then to clear the MPN cells from the body. So that study, that phase 1, is recruiting well, and we hope that the data might be presented at EHA this year or possibly ASH. And then Incyte also have a second antibody. So there was some preclinical data shared at EHA last year, and this trial is open in the US, and we hope will open in the UK as well later this year. And there was also some interesting preclinical abstracts shared by other companies of preclinical data. So this means experiments performed in a laboratory of an antibody targeting CALR that is basically bound to a drug. So it delivers a drug, a toxic compound directly to those cells. And other companies are also developing similar approaches to all of these. So a lot of activity and interest here. Okay, so patients with other mutations and hopefully not being left behind. There's also excitement about newer types of JAK inhibitors that are more selective for the mutant form of JAK. And there was a nice presentation, again, showing preclinical data, so experimental data generated in a lab of this newer JAK inhibitor tested in the laboratory against mutant JAK2. So it binds in a particular area of the protein that enables it to more specifically bind to mutant JAK, not wild type JAK. And I'm just showing you one of the plots here looking at cells from patients, so wild type healthy cells, and then mutant cells here. So when they test this drug at different doses, you can see that the drug is much more selectively reducing the growth of the mutant cells in green than the wild type cells in red. And there are other agents, again, already in clinical trials. So Incyte have one agent, and another company called Ajax also have an agent that they believe is more selective for mutant JAK. So lots of things to watch there. And then for MPL patients, I know this is a rarer subtype, and often these patients feel a bit left behind. But interestingly, MPN Research Foundation, who's a sister company of MPN Voice in the US, they just announced a new funding call for MPL mutation-focused research. So we hope there'll be some targeted therapies coming there as well. So to summarize, I think the data is really encouraging. The data shared so far shows that it's possible to achieve really highly specific and selective targeting, and potentially to modify how the disease progresses in patients. So we'll wait to see whether that's true. And there's also interest in mutant selective therapies coming in the near future as well. Of course, lots of uncertainty still in terms of who should be treated, when, how long do patients need treatment for, can they stop these therapies? And we look forward to, I guess, following these studies for a bit longer so that we can really start to answer some of those. So with that, I'll stop and hand over. I think who do I hand over to next? Maybe to Alessia. Yeah.
Hi. Let me try and share. Hi, my name is Alessia Khan. Just checking you can see my slides.
We can see them, not yet in the slideshow, but yeah, perfect. Looking great.
Great, thank you. Always the worst bit of the webinar. So my name's Alessia Khan. I'm a haematology consultant working in HMDS, which is a diagnostic laboratory in Leeds, and doing the clinic in Leeds and Harrogate. And today I will present a couple of really interesting trials, which I enjoyed hearing about at ASH. The first one is called Fedora trial, which some of you on the call might have actually taken part in. That was led by Professor Mary Frances McMullen from Belfast, and then Rope ET trial was presented by Professor Jean-Jacques Kaladjian in France, and that was a multi-center trial. So first we'll talk about Fedora. This was a phase 2 study to evaluate the tolerability and safety and efficacy of fedratinib combined with rope-regulated interferon alpha 2b in patients with myelofibrosis. And these were patients who had never had any treatment at all. So we know that lots of the trials combine a JAK inhibitor, which is the backbone for treatment for patients with myelofibrosis. And often this is combined with another agent, such as a trial agent. And there have been a few studies combining ruxolitinib with interferon. So this builds on that. And also it's using a JAK inhibitor called fedratinib, which is an oral selective JAK2 inhibitor. It can be used in the UK if a patient with myelofibrosis has failed ruxolitinib and momelotinib. So we can use it, but it hasn't been used in this exact combination before. We know it reduces the spleen size and improves symptoms, which are the targets in patients with myelofibrosis, and interferon, which we can use in patients with myelofibrosis at early stages of the disease. We know that laboratory studies have shown it can induce cell cycle activation in the JAK2 mutant long-term stem cells. So the idea is get the cells into this kind of cell activation, and then the JAK2 inhibitor can work synergistically. And what we want to know, if giving these two drugs together, if that gives a longer treatment outcome and better treatment outcome to patients. So we need to first find out if this combination is tolerable and if it's safe, and also to establish this maximum dose of the interferon, which can be given to each patient. And also we want to know how effective it is at reducing the spleen size, improving the symptoms, the quality of life, and patients had serial bone marrow biopsies, so to look at the degree of fibrosis and if that reduced by giving the combination. There were other exploratory endpoints of the study, which includes looking at the JAK clone size or allele burden. So the trial was 30 patients, all over the age of 18, and they had to have a confirmed diagnosis of primary or secondary myelofibrosis. So secondary can be after essential thrombocythemia or PV. All patients were JAK2 positive and deemed to require treatment. So either intermediate 2 or high risk when calculating the DIP score, which is the risk score. Or if they're intermediate 1, they had a palpable spleen more than 5 centimeters below the costal margin at the bottom of the ribs. All the patients had the blasts less than 10% on the peripheral blood or bone marrow. The blood counts had to be adequate. And this is the trial schema for the dosing. So all patients were established on a stable dose of fedratinib initially. And once they were stable on a stable dose of the JAK inhibitor, then the interferon, ropegylated interferon, was added in initially at a dose of 100 micrograms every 2 weeks, and then every 4 weeks that could be increased depending on the patient's response and blood counts. So each patient had a very individual dosing schedule, and these patients are able to continue on this therapy for a total of 2 years, and the results I'm going to share with you are just after 4 months. So the side effects are mostly anemia, which was in 23% of patients, only 8 out of 30 patients. There were some reports of eye disorders, which are reported with these drugs, 8%, which is only 3% patients, and there was some high potassium and high lipid levels. Less commonly, there was a few infections, some GI upset, bleeding. And when looking at the outcomes, one of the outcomes is whether the spleen reduced in size. And at the beginning, most patients, well, 29 out of the 32 patients had a palpable spleen. So the spleen was enlarged. And only after 4 cycles, which isn't very long, half of those patients had a reduction in the spleen size, which is a very good result. And then looking at the quality of life score, so all patients completed the MPN 10 score, which I'm sure you're all very familiar with. And you can see this is at this is the change from the baseline, and the dots represent the median, the mean change, and all of the symptoms have reduced. And this is only very early data after 4 cycles, so we expect after the longer follow-up that the symptoms will continue to improve. This is looking at the JAK allele burden. Again, this is really early data. If we do check the allele burden, we might check it after a year, but this is after 1 year of treatment. You can see anything below the line is a reduction in the JAK allele burden. So you can see out of all the patients, most patients are falling below the line, and there's a number of patients who had a very good response. So 8 patients had a reduction in the allele burden of 20% or more, and 5 patients had a reduction in the allele burden of 50% or more. So a very good initial response, and we'll really look forward to seeing longer term data for these patients as they carry on the treatment. So to conclude, telling you about the FEDORA trial, the initial follow-up shows that the combination of the fedratinib and ropeg is safe and tolerable in patients with JAK2 positive myelofibrosis, and so it met the primary outcome from that point of view. Efficacy is demonstrated in the fact it reduced the blood counts, reduced the spleen size, improved the symptoms, and the blood counts remained stable. An initial analysis shows the JAK2 V617F VAF reduced. So we'll wait for further data for that. So the second trial I'm going to tell you about is called ROPE-ET. This is a phase 3 multi-center single-arm study which is ongoing. And this is for patients with ET who are ineligible for standard cytoreductive therapy, and this is after 12 months of therapy. And this is just the initial data, and then further data will follow. So the eligibility criteria are patients with ET, 117 patients, and the recruitment time was supposed to be 15 months, but actually it was so popular that they recruited within 8 months, which just goes to show the need in this area. So these patients all required cytoreduction, but were either intolerant, resistant, or ineligible for all other cytoreductive therapies, which are listed there, and all patients had to be interferon naive. They received ropegylated interferon every 2 weeks, starting at a low dose of 100, and the total time for treatment was 36 months. So the endpoints were if patients achieved a durable, so that had to be over 3 months, hematological response. So the blood counts had to be stable for over 3 months and achieve platelet count of less than 400, white count less than 10, not have any thrombotic, so no blood clots, no hemorrhagic events, and no disease progression. And patients should not have progression of disease-related signs such as splenomegaly, and patients should have an improved symptom score with the MPN 10 score. So based on previous literature, the rate we would expect that people to achieve this would be 40% of patients would achieve these criteria. And also there are some secondary endpoints which they were looking at, and I'll just highlight that they're looking at the change in inflammation markers over time and also the change in the VAF allele burden. So really useful to see the dosing schedule. Patients started on a relatively small dose of interferon, 125 micrograms every 2 weeks, and then that could be increased after 3 months to 250, and then up to 500 after 6 months. And these results are after 12 months of therapy. So the patients were recruited from 29 centres in 10 European countries. 132 patients were included in the analysis, and only 14 patients discontinued, and only 3 of those were due to adverse events, which is also useful to know, as people often think interferon's not well tolerated, but that shows it is. So this is the dosing schedule of the patients, and what's interesting about this is that by month 9 to 12, a lot of patients were still on the low dose. So it shows the nearly 40% of patients remained stable on a dose of 125 micrograms, and then a third of patients required 250, and only 23 patients required the highest dose of 500 micrograms every 2 weeks. So the trial did meet its primary efficacy endpoint of 48 patients showing this durable disease response. So the blood counts were stable for more than 3 months, and there was no disease progression, and the symptoms were controlled. This is the platelet count, so you can see it goes down really nicely over the 12 months. And another really important factor was that the hemoglobin remained stable over time, and so did the white count. As many of you will know, if you've taken hydroxycarbamide, if you increase that too much to control the platelet count, often the white count can fall, but this didn't happen with the interferon. So this is the disease-related symptoms at 12 months of treatment, and you can see that 2/3 of patients had an improvement of symptoms, and a quarter of patients had stable symptoms, which really is the majority of patients, and really encouraging. Only 12% of patients had worsening symptoms, so really well tolerated drug in the context of ET. So the adverse events were things like headache around 15%, liver disturbance in the liver tests around 15%, but probably lower than we expect, and flu-like symptoms in around 9%, but they rarely required patients to withdraw the drug, only 2.3%, which is only 3 patients discontinuing treatment. So to conclude, this is a really important study, as although we've used interferon for many years for patients with ET, there are not that many formal trials demonstrating its efficacy. So this is a really important trial showing that interferon gives a durable disease response, that it can normalize the blood counts and symptoms, and the patients showed no progression in 88% of cases, and that it's safe and well tolerated. It appears to be a favourable therapeutic option for patients in ET who cannot tolerate other therapies. And I think that's everything, so I'll stop sharing and I will hand over to Priya.
So hi, I'm Priya. I'm a consultant hematologist. I'm based at Guy's, and I will be giving you a brief update on PV from ASH. So hopefully my slides are moving along. So I just want to give a bit of background. So I thought I'd start with a little biology lesson, and we'll talk about hepcidin. So some of you may be aware, so hepcidin is quite an important enzyme that plays a key role in regulating iron levels, and you may be wondering why I'm talking about this. The main thing about this is that if you're people who are normally anemic, hepcidin levels go up and they enhance your iron absorption. But if somebody has very high red cells, so for example in PV, what should normally happen is that hepcidin levels should be low and they should be sort of limiting the iron absorption. But in anyone who's got PV, iron metabolism is completely dysregulated. And the other part of this is that anyone who has PV, they end up developing an iron deficiency anyway. And this can be because of having venesections. It could be through GI loss. It could be dietary as well, just generally from your condition. And so this can actually compound how you're already feeling. So some of you may have fatigue, and it could be fatigue from MPN, but it can also be fatigue exacerbated by low iron. And so a few years ago, there was some interest in developing a mimetic or mimic of hepcidin to see if we could try and rebalance iron metabolism. And this is something that was a big focus at ASH, and this was an oral abstract that was presented looking at the phase 3 Verify study, looking at rusfertide, which some of you may have heard of. So rusfertide has been around for a couple of years. It's a hepcidin mimetic, and it's basically given as an injection under the skin on a weekly basis, so quite similar to Interferon. And what this study was looking at was comparing rusfertide versus a placebo, plus your standard of care. So what they mean by that is your cytoreduction with like hydroxycarbamide, for example, for PV. And anyone who was enrolled, just anyone with PV whose hematocrit wasn't well controlled, despite the standard of care, and they were having frequent venesections. And these are people where either they're having 3 venesections, within 24 weeks, or they're requiring more than 6 venesections in a year. And so there were actually 2 parts to the study. So there was part 1A where people were randomized to either having rusfertide or the placebo, and this was up to 52 weeks, and that was part 1A. And then part 1B was taking it from 52 weeks onward, where basically those who had been on placebo would then cross back over to rusfertide. And these were the results that were presented. And as you can see here, the key things were that quite a large proportion of patients, so over 60% of them on rusfertide, had quite durable control of the hematocrit. And this was similar, is around 70% of patients in those who crossed over. The main thing was that actually there was a lot of symptom improvement, so the fatigue improved. And interestingly, they did look at people's iron profiles and they noticed their ferritin levels, which is one of the markers we use, had actually normalized in patients. And the most important thing was that the safety profile was pretty acceptable. So unsurprisingly, probably the most common adverse event was people getting a little bit of injection site reaction. So, you get a little bit of a rash and a bit of irritation where you inject, but that was very, very manageable. And some people still had a bit of fatigue. So I think the main conclusion was this, that actually, rusfertide could potentially help reduce the need for regular venesections and could be associated with better control of the hematocrit. And again, this is with the combination with other drugs. There were some other adverse events, which I will discuss a little bit later, but I'll come to that quite shortly. I'm just going to very briefly talk about this. So some of you may have heard of bomedemstat. So there's been a lot of interest in this drug. It's a LSD1 inhibitor. So basically works on sort of the epigenetics of working on the genetic level. And it's been looked at in other disease areas, particularly ET and myelofibrosis. Ryan et al. Presented updates from the phase 2 study where they looked at this specifically in PV and in people who are basically resistant or intolerant to cytoreductive treatment, so namely hydroxycarbamide. It's very early data. It was a very small number of patients, and the primary endpoints were really focused on safety, and the secondary endpoints were looking at sort of symptom control. And I think overall, what it was found was that actually the majority of patients, there was good improvement in the hematocrit. So I think it was 20 patients in total, and 19 of them had a reduction in the hematocrit to less than 45. But it also impacted the other blood counts, so the white cells also dropped to less than 10. and there was improvement in symptoms as well. There weren't any significant adverse events. So I think the key thing were low platelets, but this didn't cause any bleeding, and there wasn't any reported major thrombotic events or any transformation. But obviously, this is very, very, very early data. This is quite a novel drug. It's not widely available, but I just thought this was quite interesting to present to you guys as a sort of alternative pathway to control PV in people who may not be able to tolerate other therapies that we have out there. So Alessia's already covered this, but I thought I would cover this as well. So ROPEG, so obviously you've seen that there was a big study of ROPEG in ET, but it's also been looked at in PV, and this is by Barbui. And this was actually a poster, and this looked at Besrami-Pass. So this was actually real world data for ROPEG interferon in PV patients across Italy. And they were basically collecting data for people. I think it was over 200 patients, of which quite a large majority were on ROPEG for over 6 months. And you can see here around 2/3 of them were able to escalate quite a big dose of 500 micrograms every 2 weeks. About a third of them, however, remained on quite a fixed dose, which is around 100 micrograms. And this might be quite familiar to any of you who've used ROPEG. The key thing here was that resection rates dropped by 50%, unsurprisingly. And actually, discontinuation rates, you can see here, 24% of people had to stop in the first year, and about 33% in the second year. And mostly that was because of, mainly sort of a little bit of liver toxicity, and also injection site as well. There weren't any major reports of psychological impact from this interferon. But I think overall, it was very encouraging and generally well tolerated. I'm shamelessly plugging here, and we do need more UK real-world data. And some of you may be aware we're running a patient survey for all of your experience with interferon. So I thought I'd use the opportunity to highlight this here. I think Alessia also mentioned that obviously from the study, one of the toxicities is liver related. And I thought this was quite interesting. So at the bottom, I've just put in a little abstract from the Austrian group. So actually, they presented data looking at ROPEG in the context of pre-existing liver disease. And they had a median dose, these patients, it was quite a small group of patients, and they had a median dose of 200 micrograms every 2 weeks. And what was interesting is actually the rates of hepatotoxicity was quite low. I think it was around 19%. So annoyingly, they didn't actually specify what type of liver disease the patients had, but I think that was quite good to see that actually, regardless of whether you have any sort of liver impairment, you could have ROPEG-related interferon, and it didn't cause any major impact in that aspect. A few other more broader themes I would cover. So there was, from a sort of sciencey perspective, John Mascarenhas had a nice abstract looking at how bone marrow fat patterns might correlate better with how well patients are responding to interferon. So this was quite an interesting study, I thought, and what they found was actually if there was an increase in the amount of bone marrow fat that correlated with lower JAK2/VAF levels, but obviously this is very, very small numbers of patients and very, very early data. So it'll be interesting to see how this could be used later on. As I mentioned earlier, in the rusfertide, there were some additional adverse events, and I think the key thing was there were skin cancers that were picked up in a couple of patients, and this might be something you guys are aware of. And there's actually another abstract from Naveen Pamaraju who presented also long-term data of rusfertide, and this is for people who'd received it for a median time of 3.8 years. And interestingly, what they found was that a small proportion of patients, even before starting rusfertide, when they did sort of a general screening, they picked up quite the skin cancers in around sort of 8% of patients. So I think it's just highlighting the importance. I mean, obviously, a lot of these patients would have had prior therapies, including hydroxycarbamide. So I think it's just something to highlight the importance of regular skin exams and PV care. And then finally, I thought this was another interesting point, was there's a lot of real world data showing that actually, and I think this is more relevant to the US, but community treatment centers had higher thrombotic events and less use of advanced therapies compared to the academic centers. So I guess showing how we need to probably, there needs to be a bit of work done there. So my top takeaways. So I did say it's going to be very brief. So long-term rusfertide is, showing pretty durable hematocrit control. So it could be something that's coming that's of interest. Bomedemstat is showing promise, but again, this is very, very, very early data. Biomarkers are huge areas of active research. I mentioned the bone marrow fat cells and things, but JAK2/VEF obviously is a big interest, and there's still real-world care gaps that do need to be addressed. And I think there's also a lot of interest now in looking at patient perspectives and how this is going to shape research priorities and patient-reported outcome measures. And I think that's it from me. So I will end my slideshow and hand over.
Hope so. So I'm just going to round off this session by talking about combination therapies. Why do we need combination therapies? And in this section, we're talking mainly about for myelofibrosis, but we also use combinations of therapies in other areas of MPN. The reasons are really outlined here on this slide. Benefit of our current drugs is good, but could be better. Sometimes short-lived, sometimes comes with side effects. So sometimes we add something else to try to get rid of a side effect. And from a new therapy or combination, what would we look for? Well, just a few things outlined here. In myelofibrosis, as Beth said, we often measure benefit in terms of spleen reduction or symptom benefit. So we might look at more patients getting that. Or we might look at different things also that Beth showed, like a normal bone marrow, big reduction in mutations or other factors. So I'm going to show you some data with combination with a BET inhibitor. And BET, bromodomain targeted drugs, affect chemicals or cytokines, particularly NF-kappa B, and target also red cell production and the megakaryocytes or platelets. So they are potentially a logical addition in MPN in general. We have been running a study with a BET inhibitor called pelabresib. And what we showed at the ASH meeting was the 96 weeks, I've highlighted it for you here, results from this study, the MANIFEST-2 study in patients with myelofibrosis. And so what we found at week 24, 6 months, was that the patients treated with pelabresib, here in blue, were more likely to get below this target 35% reduction in spleen volume compared to patients treated only with ruxolitinib. And that's definitely also true at week 96. Also, that spleen benefit tended to last longer. Remember, I said one of the things we want to do is get the benefit lasting longer. Symptoms improved, but they didn't do so significantly more enough for the study to have been deemed a success. Another way of looking at it would be to look at the patients who had both spleen and symptoms. So when we look at that, we look at this overlapping two circles. And here you can see patients on the combination at week 96. Sorry, my slides have decided to move on their own. 31% of patients had both spleen and symptoms. That was double the number in the control arm. And this is something we've seen consistently in this study. We also have been looking at the bone marrow. So you've had a look in bone marrows already. This is a normal bone marrow, normal number of cells, normal amount of fibre. This is a patient with myelofibrosis, very disordered, irregular, very deep black lines. This is reticulin, and then the blue here is collagen, which is a different type of fibre. On the study, we saw that more patients treated with the combination had reduction in fibrosis and less had worsening than the patients who were treated with ruxolitinib on their own. And we also saw other benefits. I'm not going to show you that in the interest of time, but we saw reductions in abnormal megakaryocytes and improvement in red cells as well. There's a lot of interest in allele burden changes, and in this study, all of the different driver mutations and for the overall burden of mutations, there was a subtle improvement for the combination compared to just ruxolitinib on its own. Side effects are very important for drugs And you've heard us talking about this already. This is a different way of showing them. So this shows you the number of patients on the combination that have the side effects on the left and on the right for the single arm. This is really trying to keep me on my toes, this computer today. But you can see that in general, the distribution of side effects was similar, but there was less anemia of higher grade with the combination, but more alterations in taste, for example, which is something we do see with pelabresib. And the number of deaths on study was few, and it was not different between the two arms. Where next, with this combination and other combinations, we are collecting more data. So these patients, we should be really grateful to them. This is by far the biggest study where we have continued patients on their allocated treatment. We might see some approvals. We might see another study for pelabresib. And there are other combination studies that are coming along through the pipeline. Other things that we saw at ASH for myelofibrosis, there were other JAK inhibitors. There's something called a JAK and a ROC, I kid you not, ROC inhibitor, plus another BET inhibitor. And then there was another JAK inhibitor combined with a different agent. Thinking about other drugs, there's also this drug called elritercept, which is a bit like luspatercept, which is a drug that is available in some countries to target anemia. What we showed was some molecular responses with that drug, and we're expecting a big study to open soon, including in the UK and probably all of your countries later. There are other drugs coming along to look at anemia. I'm not going to show you all the data. I just want to show you how busy and how much new information there is coming through. And then also a study of momelotinib, which is a different JAK inhibitor in combination with something called nuvisertib, which targets something called PIM. So all in all, I would say, just as we move to questions, it was a very busy ASH. We all came back pretty tired and jet lagged. What we should look forward to this year is more data on CALR therapies, more data on the other JAK inhibitors and drugs targeting MPL mutations that Beth spoke about. We expect results from trials with selinexor, which is another combination drug. We're expecting to see that at the European Hematology Association. Luspatercept, a drug targeting anemia. Watch out for news coming from MPN Voice, where you can catch up on other research and trials, and also the other advocacy groups that are present from across the world. So MPN Advocates Network and Global MPN and the MPN Research Foundation. We all work together to bring this information to you. And so now I want to ask my colleagues to join in with questions and discussion, and I'm going to stop sharing. I know that sometimes these slides are very data heavy. I can see that Priya, thank you very much. Priya, you've answered Pilar's question that was in the chat. But I'm going to ask my colleagues to turn their cameras on. And we're going to try now to answer some of the questions that you submitted online. Thank you very much for submitting them. And Beth, if it's okay with you, I might come to you first to pick a couple of questions, read them out, and then come ahead. But if you do want to put an extra question in the chat, Gregory, I can see you've asked one. We'll come to that one. But Beth, over to you.
Yeah, thank you for the wonderful questions. Actually, really fascinating questions that people have put here, submitted prior to the forum. So I'm going to merge together a few questions because several of them were on the same theme, and actually a lot of the questions are around JAK2 targeting. So the three questions that I'm putting together, one was, can we use these types of therapies that I showed for CALR for JAK2? Could there be a clinical trial for editing JAK2? And how can we help the immune system target JAK2? So to put those questions together, one thing I'll say is, many of you will know this, some won't. So the JAK2 mutation is much more hidden than the CALR mutation because it stays inside the cell. So it's harder to target with antibody therapies or cells which only look at, or therapies which only look at the cell surface. But of course, our immune system does present abnormal proteins to our own cells, do present that to other immune cells. And some of the trials that were done last year included a trial of a vaccine, which was being run both in patients with mutant CALR, but also patients with JAK2 mutations. So the study that was done a couple of years ago that some of you guys will have participated in, that the outcome of that study was shared at EHA last year, and a publication has now been submitted. So that was a vaccine looking to basically reveal or increase the amount of immune cells in the body that see and recognize the JAK2 mutation or the CALR mutation. And that vaccine was being given in combination with another drug that basically wakes up T cells in the body, so activates them. So unfortunately, that study actually had found no evidence of clinical benefit and was closed early. So that would really suggest that we need to really develop newer methods, better vaccines, and other sort of agents to use in combination with vaccines in order to really try to help the immune system target JAK2. The final thing I'll say on that is that actually other therapies already in clinical use, such as interferon, one of the ways that interferon works is actually via altering how the immune system is helping combat or control an MPN. So JAK2 harder, but there are certainly trials in this area. And then the final trial that I didn't have time to talk about, or the final study that was a really beautiful study presented at ASH, was of genome editing, which I think has led to the question that's been put in the chat. So I tried in the presentation just to focus on data that was shared that was clinical data, but there was a really lovely study done to show that you can actually edit the genes within cells from MPN patients and remove the JAK2 mutation. And that looked like a really wonderful approach when tested in cells, in a dish, and also when tested in some mouse models. So that is a really exciting proof of concept, really. But of course, it's quite a difficult approach, therapeutic approach, to then later translate to the clinic. But those are really great questions. So I'll stop there, but I can come back to other questions later.
Okay, great. So Alessia, maybe we'll come to you now to pick some of your questions.
Thank you. So thank you for the fantastic questions in advance. The first one I've picked out is that I have been injecting pegylated interferon alpha 2A since my diagnosis, JAK2 positive in 2023. How likely is a full remission over time? And thank you for the excellent question. I think we'd all like to know the answer to that question and if we can achieve that. And I think over time we will understand more about whether we can achieve a remission if we're able to do a trial to monitor the JAK levels, for example, every year. If we can, over time, for long enough, demonstrate that the JAK2 level goes down, then we can try to understand if we can get into that position to achieve a full remission. And in the trials and a few patients so far, we've already demonstrated some nice responses to treatment. So hopefully we'll be able to answer that properly over time. And there's another question about how is JAK2 correlated with the cell production from bone marrow, i.e. increased platelets or other changes in the blood cells. So basically, as you've seen from some of the slides and the talks, and to understand that JAK2 is a signaling protein that we have on all of our cells, and it helps the cells to talk to each other to produce the blood cells that we need. And we have JAK2 on a lot of our cells in the unmutated state. So how I explain it to patients is that JAK is like a light switch, and it can turn on and off when we need more cells or when we have enough. And when it's mutated, it's like stuck in the on position, so you're always making more cells. So when you have that clone with a mutation, you're always making lots of additional blood cells which you don't necessarily need. So that's how it correlates with the blood cells. So I don't know if you want to move on to.
Priya, do you want to?
Yeah, I can. Again, thank you for excellent questions. I'll see if I can. So I thought I might focus on not on the holistic side. So I will start with I think one person said diagnosed with ET since 2014, taking hydroxycarbamide aspirin, eating healthily, walking daily, how to tackle fatigue. I think fatigue, I'm sure a lot of you are aware of this. Claire Woodley is going to kill me, but Claire Woodley, our amazing AMP, has done a lot of work on fatigue. And actually, we are fortunate at GSTT that we have a dedicated fatigue services. So this is with a physiotherapist as well as an occupational therapist to do sort of exercises and strategies with our patients, and they find this really, really helpful, as well as looking at common things like sleep hygiene and things like that. I think fatigue can be quite multifactorial, so it may not always be the MPN. So if somebody says to me, "Look, I'm feeling much more tired," I'm not myself, I do always check their vitamins just to make sure I'm not missing something else that could be contributing. But yeah, I think in terms of fatigue, I think it is a challenge, and I think we do need a sort of multidisciplinary approach to this. But definitely, I would say that Claire is doing a lot of work on this. So hopefully, we will get more information, and we do have sort of fatigue module at through our Cancer Academy at Guy's, which we hope will be able to support MPN patients in the future as well. I think I'll do another one is again focusing on symptoms. Is somebody was talking about being asymptomatic for MF. Sorry, I can't get my words out. But experiencing menopausal symptoms now, and I wonder what impact HRT would have on me. So funnily enough, Claire Woodley and I are actually doing some work looking into this. So shameless plugging, sorry, Claire. And so essentially, we do recognise that a lot of our female patients, when they're going through menopause, menopausal symptoms are very, very similar to MPN, and it may not be easy to distinguish between the two. So actually, we've done a recent sort of patient survey for our own patients, and we found that lots of us are not recognising this early enough. Also, the challenges are with HRT, and some GPs, I don't know how many, or primary care physicians, wherever you are quite reluctant to prescribe HRT because they're worried about the thrombosis risk. So we're always sending letters saying, please give it because it will have an impact. So I think what I would say is, HRT is not, excluded in people with MPN. And I think if you are having symptoms, do discuss it with your MPN consultant because it is important for us to know, and maybe we can provide further support or help for this. And then I will try and squeeze in one more question around symptoms, and I seem to have lost it now. Oh yeah, sorry. So for people with early or mild myelofibrosis, what are the most effective strategies for monitoring symptoms? That's an excellent question. Especially I think for people who not on treatment, and I know everybody always comes to me and says, what do I need to look for? And I always say to the person in front of me, you know your body better than me. So you will know when something is not quite right. So we do use MPN10, which can be a good way to track symptoms. But I would say that I always defer to the patient in front of me and say, if you feel like something is not quite right, don't hesitate to speak to one of us. And I think I will hand it over to Claire. So,
Okay, that's perfect. Thanks for keeping your questions coming into the chat. We'll keep having a go at answering them. So I can see Werner Zinkland on this call, and I see you on the camera, Werner. Hello. So I saw you asked a question which is really relevant to lots of patients with MPN. Is there research going on how to deal better with itching? It's still a terrible condition for some patients. I totally agree with you that itching is a big problem. And the answer I have is I'm not aware of very active research, but I do know that one of our lovely haematologists in France is married to an expert dermatologist, and they are looking into the biology. But itching is really difficult. Do have a look, though, not you in particular, Werner, because I know you're very clued up, but do have a look at some of the top tips that are posted on different websites about how to deal with fatigue. As Priya was talking about, there's some top tips on the website and also some tips on itching. but no new research that I'm aware of, but hopefully something coming. Then there's a question on a drug called ASTX727. Is it considered viable for MPN patients who've progressed to MF? I talked to this question specifically because there was very nice data at ASH from colleagues in the UK about using this drug, which is a drug similar to drugs that we use for myelodysplasia or acute leukemia in a condition called chronic myelomonocytic leukemia, or CMML. At the present time, the wider availability of this drug from Aztecs isn't clear. In the UK, this is the drug that we've been looking at trying to use for patients with leukemia or aggressive MF. But I don't think we have access to it yet. I'm looking to Beth for that as well. Then there were a couple of questions on GLP-1. So that's drugs like Wegovy, Mounjaro, Ozempic. I was asked that question at a satellite session at ASH. Do we know anything about using this in patients with MPN? We had a thought actually about sending out a survey on that. Number one, I think if you do want to go on these drugs, be clear about side effects and do talk to your haematologist. Do let the prescriber know that you have an MPN, but in general, it would not be a contraindication. If you have diabetes or you are overweight, these drugs can help you, but they only help you if you also have a healthy lifestyle, etc. And now I think I'm going to hand over to you, Beth, for a next round of questions, if that's okay.
Sure, yeah. So, sorry, I was just typing something in the chat. Yeah, so the other question I had that was very interesting is from William Rowe. So what does the future look like for ET CALR type 1 for the timing of options being available for disease management or cure? So one thing I'll say first, so this is like the golden ticket question. We're all interested in this question, I think, at the moment. So the data that I shared was really very early. It's a phase 1/2 study, and the data so far has only been analysed for the first 6 to 12 months of the trial. So we're really looking forward to seeing how people respond in the longer term. Of course, we're hoping that the responses we're seeing, particularly those molecular responses that reflect perhaps clearance of the disease from the body, we're really hoping to see that those responses are maintained in the longer term, particularly if patients then have pauses or come off and stop their therapy. But those are the moments that are really unknown questions. In a dream world, patients would have repeated courses of an antibody therapy, whether it's a monoclonal, a bispecific, these types of therapies, or even other types of immune therapies that would clear the CALR clone from the body, at least sufficiently to then give them a long period of time off therapy. But that at the moment still sadly remains a dream. We've got prospects of it, but we have yet no real data to let us know that that's possible. So this is one thing we're all thinking about. And of course, if we're able then in the future to have drugs which are non-toxic and effective, then these drugs may be used a lot earlier in the disease than perhaps we're currently treating patients with CALR, if we can show that they do have an impact on overall longer term outcomes for people. Of course, many patients with mutant CALR in particular do extremely well on no therapy. So not everybody would require treatment. And of course, all drugs do have some toxicities, even if that's just the fact of taking a therapy when in fact you don't need one. So yeah, really interesting question, but very hard for us to answer at the moment. Shall I hand back to Alicia?
Hi, thank you. So I have another question, which is the bone marrow cellularity. What does it tell you about your MPN progression? So this is a really good question, and we, despite all these novel technologies and AI, we still do look down the microscope at the bone marrow cellularity and estimate what percentage it is. And generally in ET, the bone marrow is usually has a normal cellularity or slightly increased. In PV, initially at diagnosis, it can be slightly increased or or more. And then towards myelofibrosis, sometimes the bone marrow can be maximally cellular or it can become very hypocellular. But the important thing to remember about the cellularity, it's not just looking at the cellularity, it's about looking at many different factors all together, putting it all together, what we call an integrated report. So we have to look at that in conjunction with the blood counts, things like the LDH, the molecular markers, which we will do on the bone marrow, biopsy, and we put that all together to give the ultimate diagnosis and whether or not a patient with MPN is progressing. So I hope that's answered that question. I don't know if I should do another one or hand over. How we
Go ahead, do another one. I
So-
just WhatsApped Priya with a couple.
Okay, lovely. So another one I have here on my list is in PV, what are the key factors that indicate the start of treatment is necessary? Is a high white count one of them? So another really excellent question. So traditionally in the guidelines, the two key factors for starting treatment in PV would be if you've had a blood clot or you're over the age of 60. But over the past few years, and Professor Harrison wrote a really lovely paper in Blood last year, it was printed, and that really describes other factors which we're now taking into consideration, such as you mentioned, the high white count, number of venisections required if that's high, if a patient has multiple cardiovascular risk factors. So it's not just there are not just these traditional factors anymore that we take a lot more things into consideration. So all those things are considered by your MPN specialist. Shall I hand over to Priya?
Yeah, that's fine. No worries. So I'm just going to go through a couple more questions. So I think one of them I hope I've answered already was discussing how long do you think it will be before rusfertide or another JAK2 inhibitor become an option for PV patients whose hematocrit isn't controlled. I think it's still some time yet. So obviously, as I mentioned, we've got the phase 3 data in America. So I think we do need to wait a little bit longer. And there's obviously other novel drugs that are going through trials at the moment. Then there's another question regarding someone who has a young patient who is 36 years old and has myelofibrosis, and is there an optimal window in time to have a stem cell transplant? So I think going for transplant is quite a big undertaking, and this is something we do discuss with everyone we see in the clinic. And I think there are obviously multiple factors we have to take into consideration. So it's not just the person in front of us, but also how the myelofibrosis is behaving as well. So I think it's not a hard and fast rule that everybody has to go to transplant. I think we do have to take everything into account and how the person is feeling in front of us, and if they have any major symptoms and what treatment they're on, whether they've got other things going on with their MF. So I hope that sort of answers your question, but I think it's quite a broad question. And I think the other question I was going to quickly answer was why is raised white cell count in ET as much a concern as raised platelets? So I think it's quite similar to what we're seeing with PV. So I think there's a lot of data to show that raised white cells are associated with an increased thrombosis risk. So I think that's why we're focusing. So if someone, we say very proliferative, so we mean all the counts are high, we try to bring them down as much as we can to improve outcomes. And that's why we look at both. So I hope that answers that question. And I think I think I can hand over to Claire, if that's okay with you, Claire. So.
Not the one by WhatsApp. Okay. All right. I can see lots of active chat in the messaging. I saw a message asking were we going to translate into Spanish. We were running Google Translate. I'm hoping, not Google, we're running a translation across the webinar. Hope it's helping for you. So an important question for patients from the UK about Best-Remi and how the campaign is for keeping Best-Remi. As a group with MPN Voice, we many of us on this call were very successful in getting Best-Remi for patients during the Pegasus shortage. We are transitioning many patients off, and we are still trying to find a way to get Best-Remi for patients hopefully we will be able to get it for patients where there's a clear need. Second, there's a couple of questions about radiation. Is there any suggestion that exposure to radiation can cause MPNs or can cause MPNs to progress to myelofibrosis or acute leukemia? Yes, we know that radiation can cause mutations or changes in genetic material, and we believe that these changes do contribute, but it's very hard if you're a specific patient to show that in your case that this is true. So I would say, whilst we could see that across many thousands of patients, and we have been supporting a study called Mosaic, looking at what causes MPNs, for an individual patient, it would be difficult to say. Next, there's a few questions about bomedemstat that you presented data, Priya, on bomedemstat, the LSD1 inhibitor. We tested it in myelofibrosis, and it doesn't seem to be tested currently in myelofibrosis, but there are 2 trials going on in ET, and we would expect to read out from one of those trials, I think, towards the end of next year. The other study for newly diagnosed patients is still recruiting. And there is evidence that this drug doesn't just work on JAK2, it does work on the other mutations. So I think I'm going to close this part of the webinar, and I'm going to end, before handing over to Nona, by coming to each of my colleagues and asking them what you're most excited about for the year ahead. Beth, I see you've been busy in the chat, but are you ready with your answer?
I'm ready with my answer. I was distracted by a very nice question there in the chat about timing of mutations. So yeah, I'll keep replying to that one in a second. Yes, I think maybe people know already what I'm really excited about. So I'm really excited about newer therapies emerging that are more selective for the MPN cells over healthy cells in the body. So yeah, that goes for the CALR selective agents, but also for the emerging more selective JAK2 therapies, and of course really excited for future approaches that might also target the MPL mutation. So that's definitely what I'm most excited about.
Perfect. Alessia?
So I'm most excited about the possibility of studying the allele burden serially, and if we can hopefully demonstrate whether that improves long-term outcome if we can reduce it.
That's great. And I know that you and Jen O'Sullivan have been asking the patient community, and I want to say a massive thank you to all the patients who responded to surveys. So we're waiting, but we're really hoping for a positive confirmation of that study, which will be super important. So congratulations to you. Priya?
Sorry, I'm going to slightly copy Beth, but actually I was very excited about the more specific like JAK inhibitors that are coming. I think they're quite exciting, and it'll be good to see how effective they are.
Okay, I gave myself the hardest job because I came to myself last. So I'm excited by all of this, but you know what I'm really excited about is how we have these tools to collect information, but I'm also excited by the partnerships in the MPN field. I'm excited by the partnerships between doctors, nurses, scientists, people like Beth who do both, patients and the patient advocacy, and you do such a great job, all of you, and also industry. And I think this is really important, and this is how we're going to make things come together much quicker for everyone. I can't get my words out properly at this time of day. Anyway, so I think we're going to close the webinar with some words from you, Nona, and maybe you can tell us what you're excited about as well.
Thank you, Claire. I am so grateful to everybody who has contributed to this evening. I think you'll agree the future is really, really bright. As a patient who was diagnosed in 1991, where precious little was known about MPNs, I feel a lot of hope, especially for younger patients being diagnosed now. But the other thing where I'm hugely excited by is this collaboration with other organizations, and as Claire said, with clinicians and with patients, because all together we have a bigger voice. And that's really important because we are such a small, relatively small cohort of patients. And so therefore our voice, if we can include everybody, will be greater, and we'll see more research, more science. And I feel very optimistic, and I'm very grateful to everybody who's taken part, and thank you for your questions. Some of them will be answered if you send them in, but I tell you one thing I would love. You are going to get a questionnaire or a form from Debbie. We would love feedback on this, because what we're trying to do is meet the needs of the patient community. And so therefore your feedback is really, really important to us. And finally, we couldn't do this. We're volunteers without donations from people who are generous enough to support MPN Voice. And if you feel like giving, it doesn't matter how little, how much, it is always very gratefully received by all of us here at MPN Voice. So a huge thank you to you all, and I really look forward to seeing you soon. And it's been such a joy, and so enthusiastic to listen to all the progress being made in the background by all the research. Thank you all, and good night.